Oh shit. Okay, I apparently didn't record myself, so let me go back through it all. Okay, so taxonomy and systematics or phylogeny. Okay, taxonomy is the way that we did it before. We classify organisms based on what they look like and what their physical characteristics were. Okay, systematics is based on the evolutionary history of organisms. So they're similar but not exactly the same. Like example, birds are actually considered dinosaurs in terms of phylogeny because they came from a group of dinosaurs. Okay, in taxonomy, they're classified as their own independent group. Okay, so very similar but not the same. We understand that in taxonomy, we understand that birds are derived from dinosaurs, but they form their own independent little group. Okay. 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 So I will skip this. Okay. All right. So the three domains. Okay. So now we're on to the three domain system where we have bacteria, archaea, and eukarya. One of the things I want you to make sure you understand is how to read this chart, right? So eukarya here, eukarya in blue, archaea in yellow. They share more recent common ancestry than bacteria. Okay, so we're more closely related to archaea than we are to bacteria. Archaea is more closely related to us than they are to bacteria. Okay, so this is an endosymbiotic theory revisited and stuff like that. Okay, so phylogenetic trees are based on a couple of different things. One, the fossils, two, the genomes. Okay, as a species, we accumulate changes to our genome over time, and those accumulated changes are known as our molecular clock. Okay, so based on comparing the molecular clock between organism A and organism B, we can tell how far apart they are. Like if we compare our DNA to DNA of a chimpanzee, we can tell we're about 6 million years apart, 7 million roughly. Okay. Okay. Yeah. This is access really old prokaryotes. Okay. Okay. So binomial nomenclature. I'm sure you guys understand. We use a genus and we use a species to designate a name. Okay. So we can classify organisms based on domain, or kingdom, if they have one, and then the phylum, class, etc., etc. Okay. Okay. Couple important definitions we have to know. A prokaryote species. Since prokaryotes don't have sex at all, or very rarely, um, they can be classified as a population of cells with similar characteristics. Okay, a culture by definition is a group of bacteria that are grown in laboratory media. Okay, a clone is one member of that culture derived from a single parent cell. So you smear it on a plate, the one cell will grow into a colony. That will be a clone. Okay, and a strain is different cells from within a clone. So I take a clone and I subclone it and stuff, they will turn into different strains. Okay, so the importance of this slide is these three groups. These three groups have exact scientific definitions. Okay, Protista is not in that group. Protista is where we put all the organisms we don't give a shit about, or we don't know enough about to classify them in groups. And so there are none of these three groups. They should be put into kingdoms of their own instead of one kingdom, from what we can tell. There should be, honestly, there should be something about 7, 8 kingdoms of Protista, if I recall. And the number might even be bigger than that based on what we understand. Okay. But yeah. So make sure you know what a protist is. Make sure you not what a protist. Fungus is, a plantae is, and an animal is. Those have exact definitions. You should know them. Okay. Okay. Again, viruses are even more impossible to classify because they only survive inside other cells, and therefore they really don't classify them very well. All right, the next thing, and something you have to understand, is how to use Berkeley's manual. Okay, so this is a manual for determining bacteriology. So I can take a strain of bacteria that I don't know anything about, and I can use Berkeley's manual and actually figure it out. So I do all these steps. Okay, so I'm here now. So I do this test. If the answer is a yes, I go this way. If the answer is a no, I go that way. If the answer is yes, I go this way, and I come up with the E. coli. Okay, so like I can figure out what type of bacteria I have based on Berkeley's manual and whether or not they're yes or no. So you do a biochemical test. The answer is always a yes or a no. If it's a yes, you go to the right. If it's a no, you go to the left. And eventually, you do the next test. You go to the right. You go to the left. You go to the left. You go to the right. So you'll always end up somewhere at the bottom. That somewhere at the bottom will tell you the answer that you're looking for, and that is what type of bacteria you are. Okay, so make sure you know how to use this type of logic really to determine what type of bacteria you are, because that will show up on your test. Okay. Okay. A lot of these things are biochemical tests, and so you do the biochemical test. You can either do them one at a time, or you can just like, screw it. I'm gonna do them all together, one after the other, and shove them into this tube right here where it does all of them. Okay, so here you see citrate positive, so that's 1. Is urease positive, and there's different colors, so 2. The phenylalanine doesn't change, so it should turn much bluer. So that's 0. So these two are 1, 2, 1 plus 2, therefore 3. This is just I don't know why that 1 isn't circled. That should be 5. That's 3. That's 2. Okay, you add all these numbers up, and then you come up with a number here, and that will tell you the bacteria. Okay. All right, we'll skip the bioassay. The serology is a science that studies serum and the immune responses in serum. Serum is plasma without the clotting factors. Okay, microorganisms are antigenic, so they stimulate the body to form antibodies in the serum, and you can look for those antibodies. Okay, in antiserum, the solution of antibodies is tested against a known serum or a known bacterium, and if they bind, then you know which type of antibody you're looking at. Okay, so here, okay, is a negative test. The antibodies are brightly colored, so if there's no negative test, the antibodies are evenly spread throughout the drop, and so nothing happens. The drop appears completely white. Here is a positive test. Okay, if the antibodies are attracted to bacteria, the antibodies will only be where the bacteria are. So the bacteria will pull down all the antibodies and cluster them to where they are. And therefore they come out as distinct dots instead of just floating around the solution. Okay. Okay. ELISAs. Okay. One of the most common tests that to test for bacteria and stuff, and eventually COVID-19 and stuff, are ELISAs. So ELISAs. Okay. Here's the ELISA method. You have an antibody. Okay. That antibody is designed to attach to a specific bacteria. Okay. So say, anthrax. Okay. I dump in the anthrax. The anthrax will react to the bacteria, so it will stay at the bottom of a while. If I dump in another different bacteria, that bacteria won't stay there. So when you wash it, it'll fall off. Okay. I then add another enzyme-linked antibody. Okay. That is again designed for anthrax. Okay. And the only way I get both of these to attach to the bacteria is if it actually is anthrax. And now I can use this enzyme to catalyze a reaction. That reaction does something to change the color of something else. Okay, it's always a color change. So it'll always, and the color change will differ between different bacteria, but there'll always be a color change. And that color change you can easily measure using a spectrometer. So the spectrometer measures the color of something. So the color is different, and that means that bacteria is there. If the color is the same as the control, that means there is no bacteria there. Okay, so that's the ELISA. Okay, Western blots. Okay. The way you do a Western blot, I hated doing Western blots, is you take your bacteria, you denature the bacteria, so now you have all the proteins in the bacteria. Okay. And your little blob, you attach it to you blot it onto a paper here, okay, on a gel, okay, and so now you have the proteins in the gel. You run the gel, okay, using electricity. So the more positive ions, more positive proteins will be pulled down. So now you have a gel based on charge, okay, and now you have like very charged particles here, non-charged particles here. And you can look at the different proteins that you have. And the different proteins that you have will tell you the type of bacteria that you have. Phage typing, so you can see the phage, what phages of bacterium is susceptible to. If there's a hole, that means they're susceptible. If there's not, that means they're not susceptible. We'll skip the fatty acid profile. We'll skip the flow cytometry. Okay, DNA sequencing. One of the easiest ways to look at DNA is how many cytosines and guanines you have versus how many A's and T's you have. Okay, if they're close, if two organisms are closely related, they'll have similar amounts of GC and similar amounts of AT. Okay, when we teach you guys, we think A, G, C, and T are just different nucleotides, but they do slightly different things inside of DNA strand. So different species will have a different GC content. It's called GC content than other strands. Okay, so you'll have either high GC content or low GC content. Obviously, if you have high GC content, you'll have low AT content. Okay, but we always talk about it in terms of GC. Okay. Okay. Even more now, we can do DNA fingerprinting. DNA fingerprinting chops up the bacterial DNA and runs it on a gel, and then you can see the different fragments of the gel. So this is a control ladder, and so you can see number 6 and number 1 matches up really well. number 3, 4, and 5 match up well. 2 and 3 match up well. Okay, so these 1 and 6 are the same bacteria. 4 and 5 are the same bacteria. 2 and 3 are the same bacteria. Okay, and this one is always the one on the right side is always control. So you can also hybridize DNA, which is not that useful. Okay, southern blots. Okay, southern blots. Okay, named after this guy named Southern. Okay, so I can take the DNA from bacteria. Okay, I can attach them to DNA probes. Okay, so if I suspect that this is my, I can put probes here that will attach to certain pieces of DNA. And so I collect the bacteria and I attach them to the probes. So now the DNA is sliced and I can attach them to the probes. Okay, and if the probes attach, okay, that means I have that type of bacteria. If the probes don't attach, it means I don't have that type of bacteria. And I can then run the probes, okay, and see which ones I have. Okay, this is made by some guy named Southern. Ironically, the Western blot we learned earlier is a joke off this. The reason why they're called Western blots is because of the joke from Southern. So Southern blots run on DNA, which is why they were called Southern blots. They're named after Edward Southern. The next technique we developed was something that uses RNA. And so RNA, we call them Northern blots. That's the opposite of Southern. And so when we, in the protein blot, we're like, okay, North and South is taking Western blot. Okay, so that's where those names came from. Okay. More and more we do DNA shipping. So you look at, you slice open the DNA, you pour them on a chip. That chip has different fluorescent sequences. If it binds to that fluorescent sequence, then I know that portion of the DNA is there. And I can look at the bacteria very easily that way. And I just pop that chip in a computer and analyze it that way. Okay. So here is the DNA chip. Okay. I can recognize the bacteria based on the coding sequence here. Okay. We'll skip fish. Okay, so make sure you understand this. So we can see how close they are to each other based on genetic sequences.
Both for research and for clinical purposes, it's desirable to have a quick and reliable way to identify what kind of bacterium is present in an unknown sample. But even under a microscope, bacteria have a much more limited set of shapes than macroscopic organisms do. Furthermore, many unrelated kinds of bacteria can share the same shape. Therefore, chemical tests are often the main method used to identify bacteria. These tests are often organized into dichotomous keys, which consist of a series of paired statements in which only one of each pair of statements applies to a given organism. Dichotomous keys provide a step-by-step procedure for identifying an organism.
Okay. Okay, so just making sure.
Dichotomous keys consist of a series of paired statements in which only one of each pair of statements applies to a given organism. These statements are often worded as yes-no questions, as shown here. By answering the questions one at a time for a given organism, the organism can be identified. Flowcharts can be constructed for dichotomous keys, showing the relationships between the different statements. Let's practice using a key to identify this unknown sample. The first question in the key asks whether the unknown has gram-positive cells. A gram stain of this unknown shows red, gram-negative cells, thus the answer is no. The key then tells us to go to step 3. Also from the gram stain, we can tell that the cells are indeed rod-shaped or bacilli. The key then tells us to go to step 4. Only bacteria that tolerate oxygen will grow in a medium exposed to air. This growth is visible as cloudiness or turbidity. As you can see, our unknown became turbid in a medium exposed to air. The key then tells us to go to step 5. Lactose fermentation can be determined by growing bacteria in a medium that contains lactose and phenol red, a pH indicator. If fermentation occurs, the pH decreases, turning the medium yellow. Gas bubbles may also form in the inverted test tube. As you can see, the unknown does ferment lactose. The key then tells us to go to step 6. If a bacterium can use citric acid as its sole carbon source, it will turn Simmons citrate agar from green to blue. This unknown did turn the medium blue, therefore the answer is yes. The key tells us to go to step 7. Hydrogen sulfide production can be tested by growing the bacterium in a medium containing iron. If hydrogen sulfide is produced, it will react with the iron to produce a black precipitate. Our unknown did not produce a black precipitate. The key tells us to go to step 8. The presence of acetoin is tested using the Vogt-Proskauer test. In this test, certain reagents are added to the unknown culture. If acetoin is present, the sample will turn red or pink. This unknown tests positive for acetoin. This leads to the conclusion that the unknown is the genus Heterobacter.
Alright, so make sure you understand how to do that because that will show up on the quiz or test or both. Alright, I will see you guys for chapter 10 to chapter 11.